All right, so now we have me. I'm Karine Miras. Let's see if I can chair myself. So this workshop is about the evolution of things, including robots. And the very long-term view is that we want these things to be complex. But things in nature, oops, it's not running. A second ago it was. Okay, back to normal. But of course, we see a plethora of diversity and complexity in nature, but that complexity is not just about DNA being able to produce a certain protein. It's about timing as well. So how much, well, of course, the question of which proteins have to be produced to matter, but how much and when, and especially when matters a lot. So in this sense, maybe I can move this a little bit here. In this sense, we can think of DNA as this large book of recipes, selecting what has to be produced when. And who's the cook? Well, at least two cooks. This keeps happening. So one of them is time, which is basically how organisms develop from embryogenesis until maturation. But another important cook is the environment, selecting what kind of genetic material should be expressed when, according to environmental conditions, either internal or external conditions. And this is what I'm focusing on today, environmental regulation, specifically the environmental regulation of phenotypic plasticity, meaning that organisms can develop different traits according to different environmental conditions during lifetime in a way that is reversible. That's called acclimatization. So for instance, there can be a change in temperature that leads to a certain trait to be expressed. And then later on, the temperature changes again and a different trait will get expressed. Now, a couple of famous idioms here. Jack of all trades, master of none, no free lunch. It goes without saying that it's not difficult to argue in favor of the benefits of plasticity. Adaptation is always welcome, but evidently this should come with some sort of cost. And in the literature, costs are defined as fitness deficits. So in practice, this could mean that it is possible to produce a certain trait plastically, but then the fitness of the individual will be reduced somehow. For instance, there are some plants that can be plastic in regard to their flowering time. So depending on what's going on in the environment, they can produce flowers or not. But the gene that is responsible, or the genes that are responsible for regulating this flowering time, is also involved with the usage of water, for instance. So they become less effective in water usage. And because of that, they might become less adaptable in other senses. So they can be plastic, but the fitness will be reduced. And there are different costs, not all sorts, but at least 5 different types of costs in the literature, ranging from the actual cost of producing the new cells for the trait to genetic costs. So why should we care about costs? Well, it's one more way of trying to explain why a certain phenotype becomes what it becomes, and therefore explaining behavior, which can be useful, of course, for natural or artificial creatures. And in the literature of biology, there's not really a consensus about costs. So some studies have found costs, other studies have found no costs, even though there are reasons. It's logical that there should be costs, but it's difficult to show for all sorts of reasons. It's difficult to conclusively show costs in some cases. And one of these challenges is, for instance, isolating effects. You have different types of costs, but in nature, you can't really remove some of these costs. They're all there and you have to accept them. But that is something we can do with artificial life, because we can customize artificial systems and just include certain effects, not really effects, but include certain mechanisms or not. So in these experiments, we were working with a robotic system of modular robots, and these robots are the artificial organisms. And we're working with, so the representation of these robots is something that has been mentioned multiple times during this workshop today, which are the CPPNs. So we have one CPPN for the body of the robot, which is a modular body, and another one for the brain. And the main message of this slide is that the way to incorporate the environmental regulation of plasticity for these robots here would be to simply input extra information to the CPPN. So those can be states from the environment saying, okay, something changed in the environment. And because of that, the gene expression of whatever would have been expressed in the body or in the brain is going to change. And the objective of this is investigating the costs of plasticity, specifically genetic costs, which means, in this case, epistasis or pleiotropy, which are gene interaction or gene reuse. So the hypothesis is that, okay, there might be a benefit. There's probably a benefit, but in certain situations, there will be a cost to the point that it might not really pay off. So the experimental setup, robots are evolving in a plain terrain. The task is simple. It's just directed locomotion. Pretty common. Robots have to go to a certain direction. And here, instead of actually changing the environmental conditions, which could create extra noise, we're controlling for it and changing the conditions simply by changing the direction to which the robot has to go. So in the first, for instance, first 30 seconds of its life, it has to go to one side, and then in the other 30 seconds, it has to go to the other side. So it's a change in the fitness function that abstracts the change to the environment. And we experiment with two main scenarios, scenario A and scenario B. Scenario A represents a situation in which plasticity is obviously useful. Without it, it would be almost impossible to accomplish what is expected from these organisms. And in scenario B, there's some gain for plasticity, but it might be too small to make it, to be worth it. And the idea was creating conditions that would allow for clear results. So that's why they're extreme. So in scenario A, a robot has to go to the right in the first moment, and to the other at the other moment has to go to the left. So they're mutually exclusive. If you go to the right, you can't go to the left simultaneously. Whereas in scenario B, they have to go right and down, meaning that if they just go into a diagonal, they kind of go right and down, a little bit right, a little bit down, and it's not perfect. Some of the path is going to be missed. And the question is this worth it? Is it worth all the effort of epistasis and pleiotropy, etc.? So the first experiment validates at least that, because if none of it worked, one could be wondering, what if this regulatory system doesn't even work at all? So the first experiment will show, okay, it does work when you really need it. And on the other case, let's see. So tested different setups. In one of them, there is no plasticity. So the phenotype is always what it is, one phenotype and one phenotype only, whatever the conditions are. Then in the other case, there's plasticity in the brain. And in the other case, there's plasticity in the brain and in the body. And the control experiment is what's called a focal environment. That's just an environment without environmental changes, and in which, of course, the organisms are not able or not even need to be plastic. So the results show that what we have here on the corner is the focal environment. And what we see is that in all cases, or more like in none of the cases, the fitness of the focal environment can be obtained. So this suggests that there's a cost for plasticity. So whenever the environmental conditions do change, it's not possible to achieve that fitness that used to be possible before if you evolve only in the focal environment. And here on the left, we have that experiment that is the mutually exclusive one. So we see the obvious benefit of plasticity in relation to non-plastic. Whereas on the other side, when they could kind of take that diagonal, we don't really see a clear benefit or any convincing benefit at all. And there were no difference. Okay, this is one example of comparing morphological traits, and we see no differences in morphological traits. And that's a definition of costs. So you don't see differences in traits. The traits are achievable, but they come at the costs of fitness deficits. So for instance, here we see the robots that are not plastic, they kind of get stuck in the middle, nothing really happens, whereas the plastic robots can go to the different directions that they have to go. Same thing when the body is also plastic. But for the other experiment, the non-plastic robots can just go through this diagonal. And the plastic robots can go more assertively to the correct sides, so either right or down. But when you analyze it more closely, you see that, well, the non-plastic ones go a bit further in the diagonal. So in average, they compensate for not going so assertively to the right or down. So what's going on here? As we mentioned before, genetic costs can happen through pleiotropy and epistasis. So in this case, pleiotropy is like when a gene is contributing to different traits, more than one trait. And in this case here, epistasis, when genes are interacting. So one example that could be happening, imagine that we have gene 1 and gene 2 interacting to produce a trait, and then we have these 3 other genes used to regulate something. So we have a good trait, and then we had a bad regulatory clause. So the good trait was stomach acid. The bad regulatory clause, put it in your eyes. That is not something you want. The acid is useful, but not in your eyes. So negative interaction. So let's say a mutation is applied to gene 2, and now good regulatory clause, and it gets expressed in the stomach. But then this gene is also affecting the trait, and now you have hair in the stomach. That also doesn't help. So imagine that you could have a very specific configuration that fixes both of the problems, both the regulatory system and the trait production per se, so the structural part of the trait. And then analyzing it closer, we see, if we look at least with the morphological traits, so we could measure morphological traits and do an analysis showing that the search space became more rugged. So it becomes something a little bit similar to co-optimization of body and brain. So, but now it's the criticality of optimizing the regulatory system and the body and the brain, not only the body and the brain. So in conclusion, results suggest that the hypothesis is true. The plasticity benefits can be undermined by plasticity costs. However, we need more work because we were working with CPPNs, which structurally are like neural networks, and it's a bit difficult to analyze gene interaction. So we can't really show that epistasis is growing and pleiotropy is growing. We can infer because genetic costs is the only cost possible, but it could be that there's some other byproduct of the regulatory system that is actually responsible for this increased ruggedness in the space that we see and for the deficits of fitness. Okay, so I'd like to thank the SPEAR project for bringing me to the conference. And if you want to read the full paper, you can find it online.